Hello and welcome to Sam Biology class. Friends, today we shall discuss about Na+K plus ATPASE. Active transport is an endergonic process by which ions and molecules are transported against their electrochemical gradient. One of the most thoroughly studied active transport system is Na+K plus ATPase present in the plasma membrane of higher eukaryotes. It is also known as sodium-potassium pump or sodium pump. It pumps 3 sodium ion out of and 2 potassium ion into the cell with the concomitant hydrolysis of intracellular ATP. Na+K plus ATPase is a p-type ATPase. It is an antipode because sodium ion and potassium ion are transported in opposite direction. It was first characterized by Na+K. It is a transmembrane protein consisting of two types of subunits, alpha subunit and beta subunit. The alpha subunit is about 1000 amino acid long, whereas the beta subunit is comparatively small and it has about 300 amino acid residue. The beta subunit is not directly involved in the pumping of ion, but it facilitates the correct insertion of the alpha subunit into the plasma membrane. In some cases, an additional smaller regulatory gamma subunit is also associated with this protein. The alpha subunit consists of a transmembrane domain that is composed of 10 helices of varied length that provide a pathway for sodium and potassium ion to transmit the membrane. This subunit also has 3 well-separated cytoplasmic domains. The cytoplasmic domains are nucleotide binding domain which binds ATP. Another domain is actuator domain which participates in the transmission of major conformational chains, and the other domain is phosphorylation domain which contains a phosphorylatable aspartate residue. ATP phosphorylates this aspartate residue only in the presence of sodium ion, but the resulting aspartyl phosphate is subject to hydrolysis only in the presence of potassium ion. This indicates that this active transport system has two conformational states. One is E1 and the other is E2. These two conformational states have different structures, different catalytic activities and ligand specificities. So Na+K plus ATPase is a primary active transport that pumps 3 sodium ion out of and 2 potassium ion into the cell with the concomitant hydrolysis of ATP. The overall stoichiometry of the reaction is like this. This figure explains the function of Na+K plus ATPase. This is the alpha subunit and this is the beta subunit. For each molecule of ATP hydrolyzed, 3 sodium ions are pumped out and 2 potassium ions are taken into the cell. The E1 conformational states ion binding site are exposed to the cytoplasm. It has high affinity for sodium and low affinity for potassium, whereas the ion binding site of E2 conformational state is exposed to the exterior of the cell and it has high affinity for potassium and low affinity for sodium. the Na plus K plus ATPase appear to operate in this manner. This is the scheme of sodium and potassium transport. In first step, E1 ATP, which acquire ATP inside the cell, binds with 3 sodium ion to form a ternary complex, that is E1 ATP 3Na+. In the next step, this ternary complex reacts to form a high energy aspartyl phosphate intermediate, that is E1P3Na+. In this step, ADP is released, and this step is dependent on magnesium ion. In the third step, this high energy aspartyl phosphate intermediate relaxes to form the low energy state, that is E2P. In this step, 3 sodium ions are released outside the cell, that is, sodium ions are transported from inside the cell to the outside. In the next step, E2P binds with 2 potassium ion to form E2P2K plus complex. In the next step, this phosphate group is hydrolyzed and E2,2K plus complex is formed. In the last reaction step, E2,2K plus complex binds with ATP and releases 2 potassium ion inside the cell, thereby completing the transport cycle. Although each of the reaction steps shown in this scheme is individually reversible, the cycle operates only in clockwise manner. The Na plus K plus ATPase generate a charge separation across the membrane, that is a membrane potential. The electrochemical gradient generated by this transport system is responsible for the electrical excitability of the nerve cells. In fact, all the cells expend a large fraction of ATP that they produce, which is nearly 70% in nerve cells, to maintain the cytosolic concentration of sodium and potassium ions. So friends, it is because of Na plus K plus ATPase the sodium concentration is maintained at a low level and that of potassium concentration at a high level inside the cell, whereas outside the situation is just the reverse. So friends, it is all about Na plus K plus ATPase. For more updates, please subscribe my YouTube channel Sam Biology Class, and also please share your comments. Thank you.